Hello, everyone. I'm Eva Tosco-Herrera. I'm currently a PhD student in the doctorate program of Medical and Pharmaceutical Sciences at the University of La Laguna. My thesis director is Carlos Flores, and I'm here to present to you our latest work, which is called Causative Variant Prioritization from whole-exome sequencing patient data, a benchmarking of bioinformatics tools. So let's start. Next generation sequencing is essential to accelerate and to get reliable clinical diagnosis. Whole-exome sequencing focuses on finding and capturing the exome, which is 2% of the human genome, but it harbors more than 80% of pathogenic and causal variants of Mendelian or rare diseases. This means that we can read the protein coding regions of the genome, which has a great potential for clinics, since the search for the cause of a disease is focused on regions of interest instead of the whole genome. However, a standardized and detailed protocol does not exist yet, and this is crucial to get reliable and reproducible results. Whole-exome sequencing generally captures approximately 100,000 variants per sample, and any difference in data management may drastically change the results in the end. The aim of this study is to contribute to the development of this desirable protocol by evaluating open source tools to accelerate this diagnostic process and to reduce its cost. So for this, we selected a few bioinformatics applications to be evaluated with real whole exome data from 62 patients in total with known genetic diagnosis via other tests. A few parameters have been considered regarding this software related to their use, their reliability, and complementary options, such as output format options or HPO usage, where phenotypes can be indicated to guide the software in this process. We used proportion test to prove statistical significance in software comparison using RStudio. The whole exome sequencing process was conducted on an Illumina HiSeq 4000 sequencing system, and variants were called with 75 base paired end, following best practices on human genome 19. The overall results show that the causal gene and variant from each diagnosis was correctly prioritized by the software in 44 cases out of 62 cases in total. This is probably because their causal variants were correctly captured through a high number of reads, which we call coverage. High coverage variants are ideal for this, often correctly prioritized by the software and can be visualized in intermediate files, as shown here. But due to knowledge limits and technical limitations, the results did not show the correct causal variant in 14 cases. What we see here are examples of high coverage variant visualization in alignment files. We can call them BAM extension files. We can see here how optimal covered variants are visualized in a local genome browser, such as IGV, which is short for Integrative Genome Viewer, where reads are correctly aligned and clearly show deletions, as seen here, which means a nucleotide loss, substitutions, a nucleotide change in the middle, and insertions, adding of new nucleotides, as seen in the right image. However, when variants are not optimally captured, the visualization is certainly different. Causal variants in low-depth regions, as seen here, or variants on intronic or distant splicing elements cannot be detected as well. The same happens with long insertions and deletions that contain more than 50 base pairs. And this is known for whole-exome sequencing. This is a known technical limitation in the capturing process. The usage of an alternative reference sequence in a case specifically where the causal variant is in the mitochondrial DNA was another source of failure, as seen in the right image. So a mitochondrial causal variant may entail problems with the used reference sequence where variants can go unnoticed. This is something to keep in mind when working with this kind of data. The p-values in this table here show that Exomizer has proven a significantly higher success rate in all situations, even if we look at the first place in the first column, the first 5 places in the second column, or the first 10 places in the results, as seen in the third column. Exomizer is an open source software, which is fast, free, and currently available online. However, results from AMALI, as seen here, do not differ significantly from Exomizer if we consider a 0.01 as the level of significance. AMALI is also free open source software, but is shown as an available and user-friendly website. The latter option, AMALI, could be a less fitting solution for high throughput analysis and automatization, but it could be easier to use by clinics and clinicians or non-programmers for signal diagnosis. Also, HPO terms have proven to be an expanded system to code phenotypes because Exomizer and AMALI allow HPO terms as their input data. They hold a very relevant and specific role in causal variant prioritization, since this software showed more accurate results when this phenotypic data was allowed. So to conclude, we can say that Exomizer showed the highest success rate. It's proven its value by showing the highest success rates, probably because it leverages heavy and rich databases, but also because HPO terms can be included as input. The failed prioritizations reminded us that, well, technical limitations exist, and that the inspection of intermediate files is crucial for confirmatory purposes. So thank you so much for your attention, and I'm glad to answer any question that you might have. Thank you.
Thank you so much, Eva, for a wonderful talk. So, yeah. No questions here, please. So, if anyone wants to ask a question, you can raise a hand and we can take it live. So, yeah, we have a question by Dr. Yana, and she asked that, are you prioritizing variants involved in complex disorders as well?
Well, thank you so much for the question, Dr. Yana. In this cohort of patients, we are not, because, well, these tools are usually more specialized in Mendelian and rare diseases. So we wanted to try them out with a cohort of patients with rare diseases or certain types of diseases that have variants from a certain gene or something like that, because we are currently trying to use Exomizer, for example, with complex disorders to see what gets out of there. So we're currently on it, but this was not the case. And the cohort of patients with complex diseases were it was after this. But thank you for the question.
So another follow-up question by Dr. Yana: do you expect performance to be similar or different?
Well, I don't think I have an opinion about that. I mean, I'd rather not have any expectations, to be honest, because you don't always know what may come out of there. But I hope to see something significant, because that would be very helpful in the long run.
Okay. So another question we have by David, and he says that great talk. Will you be considering comparing annotation tools for variants identified from exome data? And he asks another question: would you be considering comparing CNV calling tools for exome data?
Okay. Well, thank you so much for the question, David. Will I be considering comparing annotation tools? Well, that was not in the plan, to be honest. It could be interesting because there are many different annotation tools out there. However, I'm not sure how could we compare these annotation tools, because each one of them gives you a certain type of information. So maybe we could try something out for the future. But that's a good point, honestly. Nowadays, the gold standard, or not maybe the gold standard, but the most expanded tool for annotation is probably Anovar, or at least that's the one that I've heard the most about. So yeah, there's that. Would you be considering comparing CMV calling tools for exome data? Well, that's a plan. That's one of the future projects for this type of assessment, for this type of software assessment. So I will certainly try to do that. I hope it goes well. So I hope to be to achieve that in the near future, to assess this type of software for CMV in the future. Yeah.
Okay, great. Thank you. We have two more questions, and it's by David from US. He says that I missed one part. How did you validate that the exomizer had the best performance overall to call the ground truth?
Well, what we did was we used whole-exome sequencing data from, as I said, 62 patients, real patients, by the way. These tools are usually validated, or at least released with a cohort of simulated patients, or exomes from a certain database that after they get them, they use causal variants for a certain disease, and they manually introduce that, and they take that variant into a healthy exome. So that's simulated data, and we tried to not do that. We decided to assess the software with real data to see how this would work out. We validated that because we tried that the 62 patients in all the tools, and we detected in which case the tool did recognize the causal variant for each one of the patients. So we had already the diagnosis, the genetic diagnosis, so we knew what was supposed to go out of each software. So success rates are the cases where each tool recognized the correct causal variant in the top place of the rankings, in the top 5 or top 10.
Okay. David, I hope it answered your question. We are running a bit late, but we do have two more questions. So this one is by Kareem. She's from Peru and she's asking, are you tracking genomic information according to the progression of some diseases?
I don't know exactly what you mean. I mean, I am tracking personally, research just to be aware of what comes out. But I don't know exactly what you mean. Like genomic information, according to the we are aware of that. Science is always changing, so we try to keep up the knowledge. At the lab. We're not doing that in any other way, to be honest. But that's always something to keep in mind because science is dynamic and researchers are always trying their best to discover new things.
Very true, truly said. So we have the last question by Azza from Saudi Arabia. She's asked, I'm sorry, are you planning for prioritizing non-coding variants or assessment of how other tools perform?
I hope to do that in the future. Not right now because I have other projects in my mind, but it will be interesting to use this type of software for non-coding variants. But especially right now, there are many more software that is specialized in exome data. Probably because, as I said in the beginning of the presentation, 80% of pathogenic variants are in the exome. So that's like the most likely place to look for pathogenic variants because most of them are there.